And it led to this study which ultimately led to what is known as the hydropathy plot. It was a real pleasure working with him and his insights which led me to many important things in proteins. We continued conversations, but before that, let me give you an outline of his career. He did his PhD with Professor Kensel Van Holt at Oregon State University. And people who are in biophysical chemistry know Ken Van Holt as one of the earliest outstanding biophysical chemists who wrote a book which we studied when we were going to graduate school. He then was doing postdoc with Don Wetlaufer, another luminary in the protein folding field, when I met him. And at the same time, when we were working on the hydropathy plot, he also published a paper, the first paper on how to define domains in proteins. And that was really a seminal work. And I used to teach when I came to join Bose Institute to students about what protein domains are. In between, he has really made seminal contributions to many areas of protein structure, including turns, ab initio predictions of protein structure, and many other fields, including, as Gautam mentioned, unfolded states, which I won't go into. He will describe some of it. It is also he is also one of the real luminaries in the field of protein folding. He went on to get his first job at Penn State and then moved finally to Johns Hopkins, where he became a chair professor. And now he is a distinguished emeritus professor at Johns Hopkins University. I think I have seen very few people with the insight that he has on protein structures and folding, and you will really enjoy his insight. With these few words, and the last thing I would say, he is the most warm, approachable, and a great human being, as well as a great scientist. You will really enjoy, and the students will enjoy interacting with him. With these few words, I invite George to the dais and deliver his talk. Thank you, George, for coming.
Thank you, Siddhartha. So as George gets ready for his talk, I just want to share with you an anecdote. He's very smart, very kind, but he's brutally honest. And brutal honesty is a very important aspect of any good scientist. I had once submitted a paper to a journal, and one of the referees tore apart my paper, and I thought the referee was wrong. So I wrote to George that, look what the referee has done. And George wrote to me, it was me. So here is George.
So do you want to turn off those mics because they feedback?
And we are still friends.
Good friends.
এখানে করে দেবে এখানে করে দেবে না অন করতে হবে এটা অফ করে তুমি এটা ব্যবহার ওনার কানে হিয়ারিং এটা প্রবলেম
So the last time I approached with this mic to that, the feedback is
okay. I'm here in case.
okay. Good.
What is this for this?
I turned it off. thank you very much for coming. Thank you, Gautam, for that introduction. Thank you for Siddhartha for that wonderful introduction. Siddhartha and I knew one another, as he mentioned, when we were much younger. And one of the few rewards about growing older is you can have had a dear friend for almost half a century. So I'm delighted to be here in India. As Gautam said, I'm now an Indian professor, at least for the moment. And it's a great privilege to be so. So today, I'm not going to talk about my own work. Instead, I'm going to try to put the understanding of science within a sort of perspective. Starting with reductionist science 500 years ago, and the motivation for doing this is that artificial intelligence, neural nets, have been so incredibly effective these days, dating back to the last couple of years. And in particular, in the area in which I work, protein folding, artificial intelligence in a program called AlphaFold has been enormously successful in being able to predict what the structure of a protein is from its amino acid sequence. And it does this in a way that is completely different from the kind of reductionist science that we've been following for the past 500 years. So it is in a certain sense at a transition point in the way we understand science and how we're going to go forward. And I'm going to try to put all of that in perspective for you. I'm going to ask a question at the end, which is, where do we go from here? I'm not going to answer the question. I don't think I can answer the question. But I leave it as an open question for you. And I'm trying to provoke you into keeping this in the back of your mind as we proceed. So with that introduction, let's go. So this seminar, how we think about science, starts with 5 centuries of reductionist science in a time in which artificial intelligence has come to play such an important role. It is, if you will, an inflection point. So science sets the stage for Enlightenment thinking. That's what I'm going to start with. And then we'll move to these large language models and AlphaFold. And I will do this from a Western perspective. I can only do it from a Western perspective. But science is pretty much a universal language. And I think you will find that it is your perspective as well. So there are times in history when a number of seemingly disparate disciplines seem to be understanding things from the kind of same central source. There's actually a name for studying that. It's called cultural morphology, but it doesn't make any difference. We recognize that there are these times. For example, at the time that the genetic code was elucidated, science, molecular biology, coming from a number of different directions, were all enlightened by just that central idea. And they seem to take their sustenance from that central idea. The time I'm going to talk about is the Enlightenment in the 17th and 18th centuries. So here are some familiar names from that period of time. It's the 17th and 18th centuries, and here you see a number of different names, all in different disciplines, so many in different disciplines, and yet they all had something in common, a kind of view of the world that was not only in common, but they directed the subsequent view of the world by what they did. So there's Boyle from the gas laws, Isaac Newton, and his name is in white because that's what I'm going to use as a paradigm today for my example. philosopher, Jean-Jacques Rousseau, and a social contract, Ben Franklin, who was instrumental in the formation of the United States. But he was also a famous, one of the most famous scientists of all during this time. Economist, philosopher, composer, oxygen, astronomer, president of the United States, chemist, once in a century chemist, Goethe who knew everything, Mozart. These names are the names we conjure with, and we study them today. But they are in disparate fields, and together they have really shaped the way that science and also understanding of things has proceeded. Much of this started with the Greeks. What did the Greeks know? So the Greeks believed that there were a number of gods, and that human activities were controlled by the gods. And if things didn't go well, it was because the gods were displeased with you. And if the things did go well, it's because you appreciated the gods. And over the course of time, that understanding began to change in the subpopulation of ancient Greece. And so Anaximander became, in a certain sense, the first really objective scientist who proposed that it's not the will of the gods, but some underlying physical principles that were responsible for the way the world worked. Pythagoras, you know Pythagoras. Pythagoras has had an incredibly profound effect on the way that science has gone. I mean, the Pythagorean theorem is something we all study in school, but it has found its application in so many different things, up to, for example, special relativity. There's the Pythagorean theorem. Aristotle, who introduced this idea of logic, scientific reasoning, and Euclid, who showed that if you start with a few axioms which you accept, an Aristotelian logic, you can conclude things that are absolutely counterintuitive. I don't know about here in the United States, we all study Euclidean geometry, not because we're going to have so much use for the understanding that an isosceles triangle of the opposite sides have the same angles, but because of this idea that if you have a few axioms and you rely on those axioms, then together with logic alone, you can begin to conclude things that weren't at all obvious, and yet you're absolutely convinced of their truth. Epicurus, who proposed that the world is made of atoms, whatever he meant by atoms, I don't know really what he meant by atoms. He certainly didn't mean by atoms what we mean by atoms, but nevertheless, that these invisible particles, atoms, which means indivisible, that these fundamental indivisible things were responsible for the way the world works. And he said that the atoms interacted with one another by love and strife. And you say, by love and strife, what does that mean? But actually, if you translate it to attraction and repulsion, then it begins to sound pretty modern. Aristarchus, who was an astronomer, used the Pythagorean theorem to measure the size of the Earth. So, a lot of this really started in Greek science, or in Greek times, ancient Greek times. But what the Greeks didn't know was experiment. They didn't believe in doing experiment. Aristotle thought that if you try to do an experiment in which you begin to perturb things in order to see how variations in, if you will, the parameters involved, change the outcome, that was going to change our understanding of things. You wanted to understand the essence of things, like Platonic ideal forms. And so you didn't do experiment. Of course, at this point in time, experiment is the adjudicator of scientific truth. We must do experiment, but Aristotle didn't believe that. So a kind of humorous example is that Aristotle thought that men have more teeth than women. It'd be pretty easy to verify that. He could have turned to Mrs. Aristotle, asked her to open her mouth, and counted her teeth. But he didn't do that. The other thing is, the Greeks didn't do quantification in the same way we do. And more than anything else, perhaps, they didn't have the kind of instrumentation that allowed us to do this kind of investigation. So, from the Greeks to modern science, it took a couple thousand years. Why did it take so long? So one reason why is because the Greeks didn't believe in doing experiment, and the idea of experimental science had to arise, but it was suppressed by the church. And there's a very nice book about this by Stephen Greenblatt, who's a professor at Harvard, and he wrote The Swerve. And The Swerve is about the secretary of a pope 1,000 years ago who was traveling around to different monasteries, and he discovered, quite by chance, the only extant copy of Lucretius, The Nature of Things. And through that book about Lucretius, that poem by Lucretius, he was able to see some trial work.
There's a slight disturbance in the, it's not your problem, some technical problem. Just wait for 1 second.
Yes.
You will take it. Go ahead.
Okay. Seems like there's always a technical problem. We've become very high-tech in these sorts of things. So this very nice book by Stephen Greenblatt, and
George,
yeah,
what they will do is they will try to put the mic little below. Okay. It might be interfering with your ear. Actually, you don't need mic.
shall we turn off the mic and see what happens?
How loud can you shout?
The mic is off. Can you hear me in the back?
Can you hear him? I cannot hear you. Okay.
Gautam always introduces humor into any situation. A very welcome humor. If I start to talk more softly and you can't hear me, please wave your hand. I'll turn the mic back on, but we'll try to get rid of these technical issues just by using something I was born with, my voice. Okay, so a quantum gravity guy named Carlo Rovelli, who's written many books, has also written a wonderful book called Anaximander on the person he considers to be the father of objective science, about 300 BC in Greece. It is an eminently readable book. I urge you to have a look at it. So who cares about any of this? There are open seats up here. Please have a seat. You can sit close. I don't bite. Be comfortable. So the reason why we care, and I think we all care, you wouldn't be here if you didn't care, is that there are a number of reasons why, but one of them is, and at least in the United States, it is highly pronounced, is the fact that anti-science rhetoric has become so dangerous these days. We have people in our Congress who believe that global climate change is a hoax. It's something propagated by scientists in order to get money. And they sell that, and it is a very dangerous proposition. Similarly, we have people in our Congress, in our governing bodies, who believe that COVID vaccine is just a way of installing some kind of monitoring system into you so that the government can watch your behavior. You know, it's nuts. But a significant fraction of the population believe it, and some people within our government espouse it. So one reason why we care is because this kind of rhetoric has become so dangerous. Science is not dangerous. It's open-ended, it's rebellious, it's subversive, but it's not deconstructionist. It's not dangerous in that sense. It doesn't deliberately mean to be dangerous. Certainly, from science, we can learn things which, if misused, would be dangerous, but science is not inherently dangerous. We value science for the same reasons that we value education. It leads to a more meaningful life. Educated people, I am sure, are able to enjoy a more meaningful life than uneducated people. And so we value that. We value learning since the time of the Greeks and before. And of course, for many practical reasons as well.
George, can you ask if people might not be hearing you? Can you hear George? Anyone?
If you cannot hear me, now raise your hand. If you cannot hear me. Don't be bashful. I really want to be heard. I came thousands of miles to India to talk to you. So I really want to be heard. If I'm not heard, please let us know.
See, the other thing you can do, George, is probably you can use that mic. If you... Hey, you might take it on, that yeah.
Oh, we're going to try this.
Yeah. Can you turn off your the other one?
It's off.
Okay.
Yeah.
Fantastic.
I tried to do this at a high level, but I'm reminded of a slightly, a story, a true story, having nothing to do with any of this, about a poet in the United States named Haas. His last name was Haas. And he became the poet laureate of the United States. And reporters asked him, how does it feel to talk now that you're the poet laureate of the United States? And he said, well, it doesn't feel any different than it used to. I'm ready to give a talk. I go to the men's room, I pee, I check my fly, I wish I were dead, and then I talk. It's kind of like that. So one of the things I want to emphasize is that you can be an honors chemistry student, like a student in senior chemistry at my institution, Johns Hopkins. And it doesn't mean that you know how science is done. It means you know how to get the right answers to questions, which are the answers in the back of the book. But it doesn't mean, I mean, you may know how science is done, but you needn't know. You need to know how it's applied, but you don't need to know where it all comes from. So this is an attempt to emphasize where science itself has come from. So what's a scientist? The grade school of definition of a scientist, in the US anyway, is that a scientist is someone who forms a hypothesis, the hypothesis is tested, and if the test is correct, it becomes a law. And that's not wrong, it's not exactly wrong, but it's certainly glib, and it doesn't describe what a scientist really does. And in a sense, it certainly is wrong. So in the United States, we have lots of people who doubt evolution, and they say, well, see, Darwin's ideas about evolution, that's just a hypothesis. It's not a law. But in fact, it's just semantics. We could equally well have talked about Newton's hypotheses and Darwin's laws. They are equivalent in the way that they're proposed, although they're in different disciplines. So what's a scientist really? I will try to put it into words. So a scientist is somebody who just mucks around trying to figure out how things work. And a theory in science can never be proven to be true. A theory in mathematics can certainly be proven to be true based on this Greek idea of axioms and reasoning. And then you can prove a mathematical idea is true in that sense. You can't prove a scientific idea is true. You can only see that it has withstood a number of tests. So how can we ever trust science or scientists? And the answer is, we understand that they are doing the best they can, and that the ideas that they have been validated by experiment. We come to understand that. And how do they do that? How do we do that in our quest for understanding? And the answer is, we have an idea, we have something we're curious about, just as Siddhartha Roy told you that we worked together on something almost half a century ago. We had an idea, and then we pursue it. And in a complicated idea, as you begin to pursue it, sometimes you find some data that seems to falsify it. And it would say, well, your idea is false. Your hypothesis, if you will, is false. But somehow we know in our gut that at that stage of the game, we don't take no for an answer. We say, we think somehow that if we just keep going, eventually it'll all be understood, it'll all be explained to us, and at that point, the thing that seems like a falsification at the moment will no longer seem like a falsification. It will be understood. It will be subsumed within the subsequent knowledge that we'll have. And we keep going until finally we reach the point where we think we finally have explained the phenomena that we were pursuing. At that point, we turn our way of approaching it by 180 degrees, and we say, now we're not going to take yes for an answer. We will do everything we can, if we were honest scientists, to try to disprove our own ideas, because we are the easiest people to fool about our own ideas. So we try as hard as we can to disprove what we've just done. So first we won't take no, then we won't take yes. And when you think about it, psychiatrists might view this kind of won't take no, won't take yes, as some kind of psychological disorder, obsessive compulsive disorder or something. But for us, it is a privileged life. So if you think back over the course of some centuries, you recognize that in the very best science, there are three more or less distinct steps. And even in some of the less transformative science, you see those same steps. And those steps are observation, which leads to pattern recognition, which in turn leads to first principles, theories, or models. And we see this again and again. So the one I'll talk about today is Newtonian gravitation. It starts with observation from Tycho Brahe, who in a certain sense was the first real contemporary scientist. And what Brahe did was he wanted to know where the positions of the planets were as a function of time. And he mapped them out by watching the planets. He used optical spectroscopy, namely his eye, and he mapped out where the planets were as a function of time very carefully and developed reams of data. That's observation, and it is careful observation. So somebody who worked for him, Kepler, Johannes Kepler, a mathematician, recognized that he could fit all of these data with his three simple mathematical laws. Then we're into pattern recognition. He recognized those patterns, but there are no principles behind that. It's just that all of these data conform to simple pattern recognition as expressed in Kepler's laws. And then finally Newton, who turned this into first principles, universal gravitation. So we're going to go through this a little bit just to establish how this paradigm works. But we see it in other things, Michelson-Morley, leading to empirical Lorentz transformations, and finally you get general relativity. Observation, pattern recognition, theory. Observe spectral lines, empirical regularities of these spectral lines, which then became evident, and finally quantum mechanics. It is exactly these same steps, these same different steps, leading to the ideas, the theories, the models that we use now to try to explain the world, the universe. So I'm going to talk, and one of the things I should emphasize is that these models are frequently counterintuitive. But we believe them because we test them by experiment. And I'm going to talk today about Newtonian mechanics a little bit. And the reason I choose it is because I'm sure that you all know it. You have to have studied it in school. I studied it in school. I didn't have the same perspective when I was studying it that I do now. And it's that perspective that I'm going to try to communicate. I got all this from a book by Richard Feynman called The Character of Physical Law. And Feynman got it directly from Newton, from the Principia. So one of the things to emphasize is that all we have really, I think, are models. We don't know what reality really is. We have models of reality. There's no, in the sense of Immanuel Kant, there really is no Ding am Sich, which we have. We have models of reality, not the thing in itself. And we tend not to believe that. When I took high school physics, Newtonian mechanics was taught as that's the way the world really works. And we believed that for a few hundred years until finally general relativity, a superseding model, and then if we didn't know it before, we realized that it's just a model. It's not the way things really work. But it is an incredibly powerful model. So the other thing I want to emphasize is that our models are mostly counterintuitive. The sun doesn't really go around the earth. I think we all believe that, but it looks that way. So seeing is deceiving, and almost all science is counterintuitive, with the sole exception of thermodynamics, which is not counterintuitive. If I have an ice cube up here at the beginning of this talk, it will have started to melt by the end of the talk. That's not counterintuitive. Although, curiously, thermodynamics, the one counterintuitive science that will undoubtedly persist when these others change, is the one that it may be the hardest one to teach, I suppose because of entropy. But in any case, experience lets us know that for some reason, it is indeed the hardest one to teach. So science as we know it, from Tycho Brahe to Kepler to Newton, and here we go in these three classical steps. And the reason I choose Newtonian physics is because I think you all know it, and it's a paradigm for modern science. So here are two of Newton's laws. One, universal gravitation, and the other, the equation F equals ma. And everything you know that you need to know about this is captured in these two equations. They have universal generality, they're taught in high school, and the idea in universal gravitation, of course, is that the force of attraction between any two bodies in the universe is a function of the product of their masses, and it goes inversely proportion as the distance between them squared. And now, when I say counterintuitive, I really mean it. So Newtonian gravitation is immensely counterintuitive. It is not something that seems counterintuitive because we've spent our lives with it. Our intuition has been molded to these ideas. In some ways, it's not entirely counterintuitive. I land on the floor again when I do that. I don't hit my head on the ceiling. So that part is clear to me, although I don't, as a little kid, I don't relate it to that equation. But recognize that what we're saying in Newtonian gravitation, there's a force of interaction between any two bodies, no matter how far apart they are. A light year apart, they're still attracted to one another by a force that's invisible. You can't see it. And if you were to try to tell that to somebody who was not educated in these ideas, they would think that you were absolutely nuts. And yet, we certainly accept it. So it's a weird idea. And if you read Newton, you recognize Newton thought it was a weird idea too, but it works. So how did Newton come by this idea? So from Copernicus, and even earlier from Aristarchus, Newton knew that the planets go around the sun. I think Newton didn't know about Aristarchus, but we know about him. So he was an ancient Greek, and he recognized this too. But we began to know this through the work of Copernicus. And so the planets go around the sun, not the other way around. So the questions are, well, if they do that, what kind of path, how fast do they go? There are lots of theories. And the idea is you would really like to understand which of these theories are correct. And so along comes Tycho Brahe, mapping the positions of the planets. That's the observation step. And it's the beginning of modern science, really. You record what you see. And then pattern recognition, it's Kepler. Here's Kepler in the 16th century, early 17th century. He's a mathematician. He's the assistant to Brahe. And from his pattern recognition, he recognizes that the planets go around the sun as an ellipse. And the sun is one of the foci of that ellipse. So what's an ellipse? Here's an example of an ellipse. So the ellipse, you can make an ellipse by just taking a couple of points, and then you attach a string to those points, and the string has to be longer than the shortest distance between the points, and then you take a pencil, draw the string taut, and continue around as long as you can, and what you get is an ellipse, which is a kind of generalization of a circle. And that ellipse has two foci, and a long axis and a short axis, and Kepler's observation was that it's an ellipse, and the sun is at one of the foci. So the orbit of a planet around the sun forms an ellipse, says Kepler. And he had two other laws: equal areas are swept out in equal times, and the time a planet takes to go around the sun is related to the size of its orbit. And it varies according to a mathematical idea, namely, it varies as the square root of the cube of the size of the orbit, where by the size it's taken as the diameter across the biggest distance of the ellipse, across the long axis. So, what did Newton know? in order to get to universal gravitation? Well, he knew Kepler's 3 laws. And so then the big question is, what makes the planets go around the sun? So Newton knew Galileo's principle of inertia. What's Galileo's principle of inertia? A body in motion keeps going in motion along the same straight line, and it goes on forever unless there's a force that keeps it from doing that. Now there's really the first Gedanken experiment, if you wish, the first really hypothetical idea. Galileo had never seen a body move along a straight line forever. He couldn't have seen it. But that's the idea. He proposed that based on what he did see. So that's the law of inertia. It goes along a straight line. And unless there's a force perturbing it, it will just continue along that straight line. Now, I wish that when I had taken high school physics, somebody had tried to say that, because I spent a long time trying to figure out why it goes along a straight line. But the answer is, it just does. You don't, that's axiomatic. So here's Newton's idea. If a planet is going in a circle around the sun, no force is needed to make it go sideways. It just continues along a straight line, tangent to that circle, to that ellipse. And it will just keep going along forever. If Galileo's law of inertia holds. But it doesn't. The planets don't keep coasting along. They go around the sun. And so there has to be a force that's perturbing them. And so, if you will, the planet is falling towards the sun. And so I want to make this as concrete as I can. So I've brought along a little demonstration. Here's my yo-yo. And now I'm going to be the sun. And what I'll do is let this planet go round and round, and you'll recognize that it's not flying out. You're in no danger. It's not going to hit you because there's a force pulling on this yo-yo that keeps it from flying out tangentially the way Galileo might have thought. And that force is, I'm pulling on it. And so it doesn't go tangentially. It keeps moving around in a circle. That's the idea. The planets are falling towards the sun. And to get that falling operation, you need the origin of the force has to be toward the sun. And so Newton was able to use that idea to show, and we won't, that the statement equal areas are swept out at equal times, Kepler's second law, is a direct consequence of this simple idea that all the changes in velocity are directed toward the sun, not just in the circle that I demonstrated, but in the elliptical case. And so we recognize, and Newton recognized, that everything is going around everything. Jupiter's satellites are going around Jupiter the same way that if there were another solar system, its planets would be going around its star. Our moon moves around the Earth. In general, everything is attracted to everything else, and the Earth pulls on the moon just as the sun pulls on the Earth. And we know the Earth pulls on things. I just demonstrated it by trying to jump up to the ceiling, but I didn't make it. I came back down. I was pulled back down. So here is Newton's thought experiment. His first Gedanken experiment. We're very fond of these. Let's just imagine something and see what the consequences are. So Newton said we can figure out how the moon falls and how it falls towards the Earth in 1 second. It takes about a month to go around the Earth. And so if you figure out how far it falls toward the Earth in a second, then you can figure out the circle of the moon, how far the circle of the moon's orbit has fallen below the straight line it would have followed if it went tangentially. Well, you can do that calculation. It turns out it falls 1/20th of an inch in a second. Now, here's the Gedanken experiment. suppose that instead of the moon, which is 240,000 miles away, circling the Earth, suppose we imagine another pseudo-moon, a fake moon. And that moon is circling at the surface of the Earth instead of 240,000 miles away. Now, you couldn't actually do that. First of all, there is no moon that does it, but also we would bump into things. But in these kinds of thought experiments, you ignore those things because you're after some idea. And the idea is, how far would the moon, if it were circling the Earth at its surface, fall towards the center of the Earth in 1 second? We know how far it does for our actual moon. How far would it be for the pseudo-moon? And so we are 4,000 miles away from the Earth's center. The moon is 240,000 miles away from the Earth's center. So the moon is 60 times further away from the Earth's center than we are. So if the inverse square law in Newtonian mechanics, the inverse square law holds, then what that means, you can do the calculation, is that the moon that's circling the Earth at the surface of the Earth should fall as 1/20th times 60 squared, that ratio, and that turns out to be 16 feet. 16 feet per second was already known. Galileo had measured it long before by dropping bodies from the Leaning Tower of Pisa. So this is one of many confirmations of Newtonian gravitation. And when you think about it a little bit, and I hope you will, it is really stunning that it works that way. You would never have anticipated that before Newtonian gravitation, that you could have done an experiment like this and found verification for it. And Newton did that. And many other examples in which these things were verified. So it was a successful theory, and like so many profound successful theories, there were so many inferences that then came from this theory. For example, it explained the tides, the high and low tides. It predicted previously unseen planets. Uranus or Neptune was then discovered almost immediately. And the speed of light in a vacuum, all of that just arose from this idea. So the take-home lesson is that the physical world in Newton's time is, in Newtonian mechanics, is like a clock. It's orderly, it's predictable, it's not random. We know that that is not quite right. But here is an incredible thing, and that is that the way that this works is commensurate somehow with our cognitive ability. We can look out at the world, and we can begin to understand phenomena, and we can begin to understand phenomena quantitatively. And that is because somehow or another, our brain is wired in such a way that together with our perceptual apparatus, our eyes, our ears, that we can begin to take in the phenomena as we perceive it, and our cognitive ability allows us to transform that into simple principles. And that is the very basis of reductionist science. Newtonian mechanics is a profound and weird counterintuitive model from the age of reason. Newton recognized, Newton, who is apparently a fairly arrogant guy, and with perhaps good reason to be arrogant, was humble about the fact that he would not have been able to do this without the information, without the understanding that came from Galileo, from Tycho Brahe, from Kepler. So now I want to make a transition from reductionist science. And here's the question. Have we now moved beyond reductionist science? So reductionist has been the very cornerstone of scientific thinking ever since Galileo. And the great scientific revolutions that have happened since then, like like relativity theory, for example, or quantum mechanics. They have been profound paradigm shifts in the way we view reality. They can account for complex reality that baffled us before, but nevertheless, the conviction that an underlying theory could account for these phenomena, that has remained intact until now. So, just a moment to tell you a little bit about the problem I work on, and then I'm going to tell you about how artificial intelligence has transformed the way we think about it. So the problem I work on is the protein folding problem. Just in brief, the protein folding problem is that if you tell me the amino acid sequence of a protein, these are the amino acid building blocks of the protein. This protein is ribonuclease A. It was one of the first proteins whose structure was known. Lots of people worked on it at the time I was entering the field. And it has 124 amino acid building blocks. And what we know from experimental work by a number of people, including Chris Anfinson, who got a Nobel Prize for it, is that in a test tube, if you will, in a reaction vessel, not in a living thing, you can take this sequence and under conditions that denature that protein, and then you dialyze away the denaturant, and spontaneously it forms a three-dimensional structure. And that three-dimensional structure is exactly shaped to do whatever the function of that protein is going to be. In this case, what this protein is going to do is to bind it. It's an enzyme. It binds a substrate. And you can just look at that and guess that the substrate will fall into that little cleft, and in fact, that's exactly what it does. And when it does, then the protein performs magic, if you will. It performs chemical reactions, a chemical reaction on the substrate, in this case on ribonucleic, on RNA, and it tears it apart. And once it does that, it is no longer held in that cleft. It's released, and then another RNA can bind in the cleft. So no cellular stuff is needed in order to have this sequence turn into that structure. It seems almost like an entropy violation. And one of the things we recognize in protein folding is that it is not an ordinary chemical reaction. And the protein goes from a disordered, unfolded state to the folded state. No covalent bonds are made or broken. Disulfides, if you will. But they are probably not instrumental in selecting the conformation, although they may be important for stabilizing. And so the folding reaction, the way it's usually represented, is we go from an unfolded state to a native state. They are in equilibrium with one another, and the change from the unfolded state to the native state is just a re-equilibration in which this equilibrium is shifted in this direction. And unfolding, the equilibrium is shifted in that direction. So I would argue that this is probably the most important problem at the interface between chemistry and biology. Just about everything that's done in us is done by a protein. So all cellular processes are mediated by proteins. Proteins, with few exceptions, important exceptions, do everything that's done in us. It's also important because the understanding of a protein and its homologs lets you begin to trace evolution, the evolution of proteins on planet Earth, which is a topic of absolutely profound interest. There are misfolding diseases, important misfolding diseases, that are just a consequence of one residue chain. Although usually a change in a single residue in that protein change does not change the overall conformation of the protein, but sometimes it does. The early example of this was sickle cell hemoglobin. One single residue change in the beta chain of the hemoglobin tetramer caused the hemoglobin to sickle. Although not because it adopted a sickling conformation, but because of the intrinsic folding of it, but rather because it exposed a hydrophobic residue where there used on the surface of the beta chain, where there used to be a polar residue, and that caused a kind of aggregation, and then the globin molecules clumped together, and then that warped the red blood cell. But there are these kinds of changes which can have a profound effect. And to understand them, we really need to understand protein folding. And finally, there is a great deal of work today, successful work, in designing novel proteins that were never produced in a mammalian system, but one can begin to design them as we understand these sort of principles. And then those design proteins can begin to do things that we'd like them to do. So we have been approaching this problem, understanding how the protein goes from there to there spontaneously for almost a century, since work of Mirsky and Pauling in 1936. And we've been using what we think are the way to approach it, physical chemistry. And although there have been many successes in understanding part of this, we have not yet, using physical chemistry alone, been able to predict how that will turn into that. It is still an outstanding problem using the kind of reductionist approaches that we have learned since Galileo. However, recently, large language models like ChatGPT and AlphaFold have had the kind of success that physical chemistry has not, strangely. So I'm going to talk about AlphaFold. AlphaFold is an artificial intelligence program that can successfully, in many cases, I'll say this more in a second, in many cases, look at that sequence and tell you what the three-dimensional structure is going to be. So let's talk about AlphaFold and how it does it, and the protein folding problem. So what does AlphaFold do? By combining physical and geometric properties, and in particular, and especially, these patterns that it extracts from the protein data bank, it does what it does successfully. And it was named Science Magazine's 2021 Breakthrough of the Year in 2021. Science looks at all science that has emerged during the previous year, and of all the sciences, all of the successes, science dubbed it the Breakthrough of the Year, and I think deservedly so. The PDB, what's the PDB? What is from the protein data bank? So the protein data bank is a collection of solved structures. It was started 52 years ago by some visionary people, and they persuaded then X-ray crystallographers and ultimately as well NMR spectroscopists and also people doing cryo-EM, once they solved the structure, to deposit that structure in this large depository called the PDB. It started with a couple of globin structures and a couple of enzymes. Today it is more than 200,000 proteins, determined experimentally. Now, the protein fold prediction using deep learning artificial intelligence has undoubtedly transformed the field by being so successful. And here are a few names that were involved in this, and there at the top of the list is John Jumper, who used to be, if you know any of these people, he used to be a postdoc, or a student rather, with Tobin Sosnick in Chicago. He has been the kind of lead person in AlphaFold. And certainly the success of AlphaFold has transformed the field. And how do we know? Well, one way you know that it's really successful is you take a protein out of these couple 100,000, whose structure you know, you pretend you don't know it, you apply the sequence of that protein, you feed it to AlphaFold, AlphaFold tells you the structure, you compare that structure with the known structure from experiment, and they match quite well. But there's another way too. There is a meeting every 2 years. It's a kind of competition, although we're not supposed to call it that. It's called CASP. CASP is the critical assessment of the structure of structural prediction. And it meets every other year. The last meeting was CASP 14. It's been going on 28 years. And and the way it works in CASP is some experimentalists who elucidate structure, like crystallographers, have solved the structure, and they know it, but they say to the crystallographer, please don't tell us what that structure is, keep it a secret, just for a little bit of time. And then they take the sequence and they tell it to people at CASP, contestants, if you will, who are anonymized. They're given a number, not a name, and they are told, go ahead and make a prediction, and then the curtain will be lifted at the end, the structure will be revealed, and you'll see how well you did. And they have an assessor, and the assessor uses something like a root mean square deviation between experiment and prediction, but it's really not RMSD, it's slightly more complicated than that, but a figure of merit. And the figure of merit, the longer this bar, the worse that they've done. And here are some of the other really top contestants, and here's AlphaFold. It blew the others out of the water. So that's how we know it has been really successful. And these machine learning algorithms can predict structure at atomic resolution, and they often can do it today in seconds. And so these methods have now solved more than a couple 100 million protein structures, all the proteins from whole genomes in places. And they're accessible to you. You can, on your dining room table, you can set up a computer like this one, and you can download any couple 100 million for free from a database. So the ability to do that has been truly transformative. How does it work? Here's the paper, and the paper is from the Nature paper for AlphaFold. John Jumper was the lead author on that, and here are the steps. And I have to confess that I don't fully understand it, but the overall idea is that you start at one of two places. You either start with a genetic database from which you get not only the sequence that you're working on, but if you're lucky, many homologs for that sequence. And you also start from the structure database like the PDB. And there are other structural databases as well. And what you do is you begin to go through a kind of cycle. So if you're dealing with homologs, you do multiple sequence alignment, and then you begin to take that information and feed it into something that's called the evoformer. I don't know exactly what the evoformer does, but it has many blocks, it has multiple neural nets, and it begins to operate on this. And what you're trying to do is minimize some kind of loss function, and you go through other steps with additional neural nets. I think there are 3 different neural nets in this whole process. And same here, you're going through something like that. And now you arrive at an end. You're trying to minimize the loss function. You recycle back again, and you go through this 3 times. And at the end, what comes out is a kind of a model that's a little bit fuzzy. Why is it a little bit fuzzy? Because unlike anything you might ever have imagined, the way that this starts is by taking the central atom in each of the amino acids for this protein that you're trying to predict and put it at the origin. Everything starts at the same point. And what you finally end up with is something that has the overall topology of the protein. And then at this last step, there's no physical chemistry in this at all, except at the last step. And there you're using a molecular dynamics approach to try to make this prettier. But even if you didn't make it prettier, the structure is clearly there. So you pretty it up at the end by making the atoms, moving the atoms to a place a little bit that is a little bit more in keeping with what you think the energy is. But it's already there before you ever do this. So no physical chemistry whatsoever. This ridiculous starting position where everything is at the same origin, and yet at the end you end up with the structure of the protein. So it uses multiple neural nets. What's a neural net? Here's a book by Wolfram called What is ChatGPT and Why Does It Work? It's a very readable book, and he's describing what a neural net is. I recommend this book if you're interested in this stuff. In the human brains, there are almost 100 billion neurons, nerve cells, and each of them is capable of producing an electrical signal of perhaps 1,000 times a second, and the neurons are connected in a complicated net. And each neuron has tree-like branches, allowing it to pass electrical signals on to perhaps thousands of other neurons, and in a rough approximation, whether any given neuron produces an electrical pulse at a given moment depends on what pulses it's received from other neurons with different connections, and they have weights attached to them. So this is a complicated, interconnected network, and we have one. And this whole artificial intelligence idea is modeled after the one we have, and ours is in our neocortex. It is the part of the brain, it's the part of the brain that has gotten enlarged with evolution. Recognize that you don't have one brain, you have multiple brains. They emerge during different times in evolution. It's surprising in a way that they all seem to work together, at least to some extent. But the largest part of the brain, and the one that keeps evolving, is the neocortex. And in the neocortex, you have a neural net. So here's a 3-layer example of a neural net, but yours is actually 6 layers. And it has an input layer, an output layer, and then hidden layer. This is a 3-layer, yours is a 6-layer, the one in AlphaFold is 6 layers as well. But you see, they're all connected with one another. And the problem with understanding the way that we understand reductionist science, and we do understand it. I mean, Newtonian gravitation took more than a century to be developed. But you can understand it in a high school class. You understand the fundamentals of it within an hour. It took a century. But you won't understand this one, at least not yet, because there are all these hidden layers. So you know the input, you know the output, but you don't understand how you got there. And in a certain sense, that's true of your brain too. You don't understand how your brain processes this information, but it does. So, one of the things to recognize is that deep learning networks like AlphaFold work in a very high dimensional space. They have billions of parameters. It was the breakthrough of the year in 2021 with billions of parameters. It does pattern recognition. It's not doing physical chemistry, and yet that works. So I want to emphasize that it's a major advance in fold prediction, but not folding prediction. It tells you what the fold is, but it doesn't tell you how you got there. And we want to know how you got there. So I belong to this working group that does coarse grain modeling and meets at Telluride every other year. And all of these people who come to this working group are working scientists. They're active. I'm active too, but I'm formally emeritus. But I'm active. There's one postdoc in my lab. It's me. But all of these people are truly active. They run big labs. And what they find is because AlphaFold has been so successful, funding from the agencies has been drying up. The agencies want to fund things that are really successful. And it's drying up at the expense of these people who are still trying to use physical chemistry. And so they asked, what are we going to do about this? So I said, well, what we should do about it maybe is to write a paper and publish it in a conspicuous place, telling what AlphaFold does and what it doesn't do, and explaining why we should keep going with what we're doing. And they said, yeah, that's a great idea, you do that. And I said, no, you're the ones whose funds are being compromised. No, you need to do that. And we went back and forth. And eventually, I agreed to do it. So here is that paper that was published a little while ago, and it is the difference between protein folds and protein folding. So the thing about AlphaFold is it recognizes the folds. It cannot know about protein folding, or at least I think it cannot know, because the patterns it's recognizing are coming from proteins in the PDB. Those proteins have already folded. They have been pre-organized by whatever the physical chemistry involved has allowed them to adopt. And when you see them, they're in their final state. And that's what AlphaFold knows. It doesn't know how they got there. There's some papers that are appearing in the literature at this point that say that they are discovering the underlying physical chemistry. I doubt it, but I call to your attention that some smart people think that what I'm saying now is wrong. So I have to acknowledge that. But these patterns are fully baked, if you will. They're pre-organized. And they were already realized during folding, and you don't know what they were. So the thing I do want to emphasize is that the way that AlphaFold comes on these patterns is they're extracted from the PDB. Now more than 200,000 proteins. Had there been no PDB, there would be no AlphaFold. So, if you will, it's an existence proof that the patterns exist. They must be there, otherwise AlphaFold couldn't extract them. So, here are a couple of analogies. One is that AlphaFold is rather like going to a movie, and you just watch the last 15 minutes of the movie. So you know how it ends, but you have no idea what the plot was in getting there. So, and a better analogy is that if you look at Mendeleev's idea about the periodic table, his compilation of the periodic table, just based on the properties of the elements. And it was sort of sketchy, but it was insightful. That is kind of pattern recognition. And then comes quantum mechanics and Linus Pauling. And now from quantum mechanics, you are able to reproduce the periodic table just from some underlying principles. This is an enormous cognitive feat. It's a triumph. AlphaFold doesn't work that way. So John von Neumann liked to make fun of people who overfit their data using too many parameters. He said that with 4 parameters, I could fit an elephant. If you give me a 5th parameter, I can make it wiggle its tail. He's making fun, and he's having fun. In contradistinction to that, AlphaFold uses billions of adjustable parameters, and in a sense, it's like a child learning a language. When a child starts to learn a language, they learn it incrementally. Pretty soon they can speak eloquently, perhaps, but they don't know the underlying rules for the language. They don't know the grammar for the language, and they needn't know it in order to be a speaker of the language. So science as we have known it from reductionist science, once again, are these 3 steps out of observation, pattern recognition, and above all, finally, theories and models. Biology hasn't quite done this because biology is harder than physics. Biology has many more parameters. It's self-modifying, it's context-dependent. Biology, over the course of evolution, things begin to change. And so you need controls for biological processes, like the kind of processes that take place in us when we have our breakfast and turn it into ATP and or the energy to think and to move. So biology is lagged behind, but recognize that some of the ideas we have really don't apply to biology because it is that complex. For example, what's the control for the speed of light in vacuum? And you say, well, that's a silly question. The speed of light in vacuum is a constant. It doesn't need a control. But in biology, we need controls for everything. So that's science as we know it. Perhaps the place that we have come to is we have data, observational data that comes from solved structures. Now, I think, although not everybody does, that deep learning artificial intelligence is like pattern recognition. And will there be a next step? Will finally we have models that are able to reproduce what we have from AI? I don't know the answer to that question. You will, I assume, be able to answer that question in due course, but in due course may turn out to be years or perhaps even decades. I don't know. It's been almost 100 years so far. So what's next? It's going to be very interesting. Please keep your eyes open, and I really want to thank you for your patience and for coming to this talk and for the privilege of speaking to you here today. And I'm happy to take questions.
So,
and I should emphasize before we begin that although I have hearing aids, I'm still quite hard of hearing. And in addition to that, that coupled with the fact that often I don't understand the local accent. So I may not be able to understand your question. But fortunately, I have a brilliant communicator who can translate English to English if that's what I need.
So you have a choice. Either you ask a question and don't ask two questions. In India, very often you will say, sir, I have two questions. So ask one question at a time, even if you have 5 questions. The other is you may not have a question, but George did end this talk with a question, what next? And I'm sure some of you have the answer. So you can say, hi George, I have the answer. So either you ask a question or you answer George. Okay.
Really intriguing talk. I just have a question that from multiple sequence alignment, going through this cycle and eventually predicting the fold, there is another approach adopted by David Jones. I'm sure you know that, and that is from mutation suppression leading to a covariance matrix, like 2 positions of a protein evolving simultaneously, and you can predict fold from there also. But it indirectly involves some kind of physical chemistry also. So since AlphaFold is completely taking the pattern recognition approach, how do you think is the possibility of these two, independent approaches being combined together?
So I think I understand the question,
Okay.
and you probably heard it. I'll use the mic. So even these approaches, all of these approaches that are not AlphaFold, but use some kind of covariation, depend upon recognizing homologs. And at this point, for a given protein, if we're lucky, we have a large number of homologs. And so the idea of the homologs that has been around for a while, but hadn't worked, is that if you look at some protein of interest to you, and then you have hundreds of homologs of that protein, that have occurred during the course of evolution. And you line them all up as best you can. You do a kind of optimal sequence alignment. And you notice that when residue A changes, residue B also changed in many or perhaps even all of the homologs. You say that there must be some kind of coupling between residue A and residue B. Seems reasonable. And then you see residue B changes coordinately with residue C. And so you begin to build up these networks. Now the reason it hasn't worked is because that doesn't always work. There's not strict transitivity like that. But about 10 years ago, Deborah Marks and Chris Sander and their colleagues, then I'm trying to remember, in any case, at an institute in New York, Sloan Kettering, when they were working at Sloan Kettering, they solved that problem using a kind of maximum entropy approach. And they're able to distinguish between A and B and C, and it's not true, and A and B and C, where they really are coupled. And then what they were able to show with no X-ray structure at all, but just sufficiently many homologs, that if they could line them all up, they could predict the three-dimensional structure of a protein. Not at high resolution, but they could predict it as well as a set of lower resolution X-ray structure. And that was a really big thing, but you needed to have sufficiently many homologs. Without that, you couldn't do it. And now others have recognized that when they're trying to use what are called residue propensities, the propensity for, say, alanine to be in a helix rather than a beta strand, or something like that. If you could couple that with information that came from these kind of homologs that are available in the database, and begin to do things like what you just described, would do better. And so now, just doing that, you can predict the secondary structure at about 80% level at best, not 100%. But it works quite well. And it is another implementation of this idea of leveraging homologs in order to be able to better understand the formation of the secondary structure of the protein. Thank you for the question.
I'm trying to give you a little background for this, answering this in a more expansive way.
There are some very smart people here. Be careful.
Yes.
I'm going to ask you hard questions.
I hope so. And if the answer to the hard question is, I don't know, I will say, I don't know. I'm smart enough at least to do that.
I have a curiosity-driven question. If I am correct, I am not mistaken, protein folding should have effect of pressure, right? Like atmospheric pressure that you define while calculating the protein structure. So in that case, let's say if you take a protein to the space, it might fold into different structure. If that is so, then how does it play out in terms of, in the case of Cluster number, or if someone wants to settle down in Mars or Moon and in the future, how can it affect the protein folding, and especially it may affect the future direction of evolution?
He's a young faculty member who joined Bose Institute.
Yes.
He's asking you to comment about protein folding in Mars, where the atmosphere is, right?
What makes, that's what he said. What basically he's saying, the question was, what happens when folding is, you change temperature and pressure?
You change the program, you change the principles.
Change the database.
My question for you, to understand your question, are you asking about proteins that can adopt multiple conformations, or are you asking whether if the folding rules were different?
Now, my question is a little broader. What's the biological significance of changing pressure if we want to see that in space, or if we want to make changes in the pressure?
Biological significance typically referees ask. What is the biological significance? You have been referring papers. No. So, George, you want to comment?
So, I'll use the mic. Okay, so for the last time I was on Mars, here's what I saw. So I'm going to give you a slightly long-winded answer to your question, but it's a visionary question, and I will try to give you a visionary answer. So, starting in 1951, Linus Pauling, before any structure was known, deduced from model making the protein alpha helix. And everybody knows the protein alpha helix. And the way he did it was he made some simple assumptions, and from those simple assumptions, he derived the protein alpha helix. It was a very successful model, and it was confirmed almost immediately because at the time, Max Perutz was doing X-ray crystallography in Cambridge, England. Pauling was at Caltech in California. And what Perutz did was to, from the alpha helix that Pauling had predicted, there should be a particular reflection. He had a 1.5 angstrom distance, and that should have led to a particular reflection. Other people were proposing models, helical models at the time. None of them would have had that kind of reflection. And Perutz then did X-ray crystallography on a couple of examples. One of them was hemoglobin, and sure enough, there was the reflection exactly as Pauling had predicted it. So it was a great triumph for model making. And Pauling was, of course, lucky because the example that Perutz had used was an all-helical protein. I mean, he might have been working on plasticine, which is an all-beta protein, and then he said, nice try, Pauling, but it didn't work. Okay, Pauling's model used very simple ideas. It used bond lengths and bond distances that came from small peptides, or just amino acids. And he knew what those were because he and Corey had elucidated those structures earlier. And he also knew that the peptide bond had partial double bond character, and the consequence of that was that the conformations that were available were simplified. It winnowed the number of conceivable conformations. Now, that is a model. It uses only a couple parameters. It's from chemistry. Now comes another Indian scientist whose name is Jayant Bengaluru. So I don't know, some of you probably know who Jayant is. So Jayant and his colleague Amos Maritan have been working for 20 years to be able to develop a model for the alpha helix and for beta strands that doesn't depend upon any chemical parameters. No hydrogen bonds, no distance, no partial double bond character of anything, just geometry. And working with Tatjana Skerbich, who was a postdoc in Eugene, Oregon, where Bengaluru is at the moment, and she's now returned to Venice. But working together, they have finally succeeded in solving this problem. They can derive something that almost exactly matches an alpha helix with no chemistry whatsoever, no hydrogen bonds. They have one parameter that comes from proteins of known structure. It's a scaling parameter. Because what they develop is a family of helices. They are all space-filling helices, and the idea is to generalize spheres. Polymer chemists have been working on polymers as spheres, and they generalize that to coins. And these coins get stacked in a particular way. No two coins can be in the same place at the same time. They are objects that are space-filling, and they generate something that is a consequence of this, which is really a space-filling helix, meaning that the coils of the helix are in contact with one another. And the only thing that they use from known chemistry is this scaling model. So they can make a really big helix out of big parts, a smaller helix, smaller. And so they use one scaling parameter, and then from that, they develop, just from geometry alone, something that is identical, almost exactly identical to the alpha helix. Now, with that background, I'm coming to your question, finally. And that is that when they solve this problem, they don't have just a right-handed helix, they have a left-handed helix as well. There are two solutions to the problem. There are always two solutions to so many problems. And we know that here on Earth, where proteins are made predominantly of L-amino acids, you have predominantly right-handed helices. And if you try to take a right-handed helix, if you try to make a left-handed helix out of L-amino acids, you get a steric clash. So it doesn't work to make left-handed helices out of L-amino acids, although you can make a little tidbit of them from time to time. But in the Bengaluru solution, which is an absolutely theoretical solution, you get both a left-handed helix and a right-handed helix. Now, P.A.M. Dirac, the great physicist, derived quantum mechanics from a different approach than Schrodinger had used. And in his quantum mechanics, for the equation for an electron, he had found that there are actually two solutions to that equation for an electron. And he thought that was by chance, but it didn't mean anything physically. And that other equation, which is now called the positron, actually turns out to be antimatter. And there is antimatter. And for every piece of matter, there's an antimatter corresponding to it. Feynman made a joke out of that. He said, if you arrive at another planet and you meet somebody and they say, we're so happy to have you here, and they stick out their left hand to shake your hand, don't take it to them. Because you know, matter and antimatter destroy one another. So, two solutions to the Bengaluru helix. What if in some other place, like Mars, it had been established with D-amino acids rather than L-amino acids? And now the predominant helix would be a left-handed helix. So these would be different rules, corresponding to ours, if you will, but it would be anti-life, not our life. And so the answer to your question is, yeah, maybe.
Okay, question, comment.
You ask a simple question. I'm sorry, I give you a complicated answer.
Okay, so first of all, I would like to thank you for this amazing talk with so much inspiration. So my question is, we talk about this reductionist approach. So that actually works well in physics, as you said. So what is the reason? I mean, what do you think would be the reason it doesn't work in biology? In the sense of say length scale. So it works in physics because of the length scale. So we have certain. So at what length, for example, what could be the length of a living being, which actually for which you think that this reductionist approach is actually breaking down? I mean, I'm just trying to naively connect the two things because this approach of reductionist, I just want to know where it scales. In the sense of length scale, if it is involved, and because I work in strings, so whether it is below Planck length, I mean, what do you think in the sense of where this approach breaks down?
So you understood the question.
So the echo absolutely confounds my hearing.
Okay, I'll tell you. So he is a physicist. He works with strings. I guess not guitar strings. His question is the reductionism that you talked about. And he says that as far as he's concerned, correct me if I'm wrong, reductionism works in physics. But he feels after hearing you, it has a problem in biology. So her question is why, and he's giving a hint. Is it a scale problem?
So my question is like, what is the size of say the smallest?
Yeah, it's a too complex question. We can answer. Yeah, understood.
Okay. So my basic answer to that question is I don't know. But the thing that I suspect is because it is perhaps a scale problem in a certain sense. Biology is lots more complicated. And it's lots more complicated because it's self-modifying. And we presume that so many of these things in physics are not self-modifying. They may be complicated. I mean, what I know about string theory is what I read in a book that tries to describe string theory, but I certainly don't understand it. But you know, the example that everybody uses is that the early Earth didn't have oxygen in it, or at least not very much. Then there are plants. Plants produce oxygen as a byproduct. The atmosphere begins to fill up with oxygen. Oxygen is toxic. Critters aren't able to survive in an oxygen environment. But over the time course of evolution, that we have learned how to not only survive with oxygen in the atmosphere, but benefit from it. We capture the oxygen, we utilize it in order to oxidize things. So when I had my Italy and sambar for breakfast this morning, I am able to oxidize it and turn it into the kind of energy I need. So I suspect that in biology the reason is because it is self-modifying in so many ways. It is so much more complex.
Thank you.
Yeah, I had a question, which is that, firstly, to modify his point, apart from reductionism, there has been a lot of emergence. Beginning with Phil Anderson. But anyway, that was not the question. Question is that, I know both Banawar and Maritain. I know both of them very well. I was not aware of this work. Thank you. I'll go back and look. But my question is that, you seem to be quite you seem to be quite appreciative of Banawar and Maritain's work, which is, I guess, more of an allostric approach. But you are not so accepting of this defined thing that, the AlphaFold thing, where they are using the geometric constraints.
What was your question?
That is the question.
What is the question? It should end with a question mark.
Yeah, the question is that Banawar and Maritain, he seems to be quite appreciative of their, where there's basically an allostric approach, right? A scaling approach. And there, no physics laws are being used. However, in the case of AlphaFold 2, they begin with geometrical constraints, right? So, it's not that, I understand that they are not using very deep physical chemistry principles. So, if we are accepting Banawar's, why not the deep?
Can I?
Sure.
So his question, okay, he's a very smart faculty member here.
I take it as a given that everybody knows.
Yes, ma'am. So his question is that when you describe that Giant's work,
Not use the microphone, the echo is.
okay. So he said that when you talked about Giant's work, Giant Banawar's work, it was based on physics. Whereas when you talked about AlphaFold, you said there is no physics. And then he adds that one of the inputs of AlphaFold is distance constraint. What he means probably is that actually AlphaFold, if you go into the details, it takes sequence, it also takes structures, but from structure what it does is close contacts, which is not really a constraint, it is still some kind of a pattern, but I guess that is what you mean by saying this.
Almost. I mean,
but really, that is a pattern, there is no physics. There is no question, there is no
but that is the point. I mean, Banauer and Maritan's work also does not take very deep physics. I mean, they are basically taking scaling.
So he says, similarly, he feels that Jayant's work also does not take into account physics at that level. So would you consider their work as much as pattern recognition as AlphaFold? Something like that.
So the idea in Jayant's work is there is no physics. It's just mathematics. It's just geometry. So I agree. There's no physics in it.
Right.
So I don't know what to say beyond that.
Yeah. I mean, so.
So do you agree with what you said?
Yes. Yes.
So it's resolved.
Sorry, I realize we've given short shrift to your question. But
Hi, George. Here. Hi. Yeah, so I'm not going to ask you any scary questions like that, but you ended your talk, which is very inspiring for very, junior people like us and who are pursuing science. It was inspiring. And you ended it with an open question, like what after AI? I was thinking about it, and I just want to discuss it with you. What I think is, let us imagine nature gives us consciousness, like in humans, and we can recognize patterns. So now we prepared other stuff like neural networks so that they can recognize patterns for us. And we are giving them automation. They are going to, maybe a time will come when they will prepare tools for themselves, those AI, and they will try to do the other stuff and leave these automated things and pattern recognitions to those tools. Maybe we are giving them too much automation, don't you think? Autonomy, rather.
Do you understand?
I didn't, so
She said, hi, I'm much younger than you. And I love the question that you posed at the end. And I have been thinking about it. And I really want to spend one whole day discussing it with you. But because Gautam is not giving me time, I just want to say, and correct me. And she said that what we did as humans is pattern recognition. Now we have neural nets that are doing pattern recognition. And maybe neural nets will make
another,
neural net.
Something different to do that work.
And then she gets lost. And then she poses
and what after that?
And then she gets lost and poses the question to you, says, what do you think will happen?
Got it.
Right?
Yeah, absolutely.
Okay. Good question. So, I do recognize that what we use is a neural net. Our neural net is I gave an example of the way we learn to speak. It is basically a neural net. We have no rules. Later we learn rules. The fact that we can also come up with rules and extract rules from these patterns is something that is part of our cognitive ability. I don't know how we got that. It's a problem for neuroscience. We are, after all, chemistry. We are chemical beings. In about the same time that Einstein was alive, was a philosopher named Henri Bergson, who believed that you could never capture the essence of life with chemistry. He said that there was the élan vital, the vital spirit, and the vital spirit was not a tangible thing. And then along came Watson and Crick and showed that it's a double-stranded helix that explains so much. But still, how did that get implemented? And then Kornberg showing the DNA polymerase. And suddenly it became really obvious that so much of this is just chemistry. Now, you're asking a question about neurochemistry, and we don't know the answer to that at this point, but I will pose to you a question. So, could we have a thought in a test tube? And after all, if we had, and I don't mean a test tube literally, I mean a reaction vessel, independent of us. After all, our brain is able to generate a thought. It does it with electrical impulses, and those electrical impulses are formed by metabolism, and there it is, and there's a complicated connection, and we get a thought. Could we have in your neural net, which you actually put together, not by artificial intelligence, by actually by actually producing something, synthesizing something, could we have a thought? And so here we have a test tube, and it has a thought. I don't know exactly how we know that the test tube had a thought, but it does. We'll just suppose that it does. And if we don't wipe ourselves out by the end of the century with global climate change, we may actually reach that point. Now suppose you have a thought in a test tube, and another test tube with a thought, and yet another test tube with a thought, and one of the thoughts in a test tube is, I'm a thought in a test tube. That's us. Could it occur? I don't know. It seems to me, in principle at least, it's conceivable.
Keep it short.
Hello, sir. I have a question regarding the prediction of protein fold. So, before that iterative process, we have this genetic sequence. So, from the genetic sequence, we get the amino acid residue sequence. But we know that the codons might be degenerate. One codon can code for multiple amino acid residues. So, how can we predict the sequence? How can we predict the structure if we are not sure about the amino acid sequence? Because suppose one codon codes for two amino acids, one with probability T and another with probability 1 amino acid.
One codon does not code for many amino acids. Many codons code for one amino acid. Change your question.
Okay, so I was mistaken, sorry.
Thank you. Okay.
Yeah, I'm a parasitologist by training, so my question more of a parasite biology. So many of the parasite proteins, like circumscribed protein of malaria parasite, then cords and flounders, antifreeze protein, many of them have a repetitive sequence. And Yerny, one of his Nobel lecture, described this like musical compositions, like it's a repetition. So how your alpha actually putting this kind of repetitive sequences that you see in the parasite system, and does it extend in your AlphaFold system?
I sort of understand, but help me.
Yeah, so his question is, how does AlphaFold deal with low complexity sequences? So where you, no, those are called low complexity, where you have repetitions.
How does AlphaFold deal with low complexity sequences? And I might say intrinsically disordered proteins. And the answer is not very well. So it doesn't, first of all, they may not have a structure until that low complexity sequence interacts with its cognate structure. And then it adopts a structure. But that process is one in which there are not examples of what it's going to be in the future in the protein database. And so AlphaFold doesn't deal with it well. And of course, the reason it doesn't is because it's doing pattern recognition from many examples. And there aren't examples of that in the PDB. Similarly, there are now some fold switching proteins, proteins that actually adopt a fold, and then something happens and they switch to a radically different fold. And there are examples of that. And although people haven't realized that previously, now there's a fair amount of work on it, and more are being elucidated. Apparently some major fraction of proteins can do that kind of fold switching. AlphaFold does not do that well either. It recognizes one of those folds if it turns out that there are many examples, but it doesn't recognize the alternative folds.
This will be the last question.
Thank you, sir. My question is, with the advent of this AlphaFold software, is it capable of determining or recognizing the modification sequence in the protein, such as phosphorylation, acetylation, methylation, and with being so much accurate with the protein structure, in the near future, will structure elucidation instruments like NMR and XRD, will it be obsolete?
It's an existential question. What could happen to experimentalists? I think this is more important. The first part was more like,
Translational modification.
Yeah. So, but I think are you an experimentalist? Yeah. His existence is at stake. This is the last question.
Well, in a certain sense, this article I wrote, this opinion piece I wrote on the difference between a protein fold and protein folding, I wrote it just for people like you. So, what happens to experimentalists? I think at least at the moment what happens is that if you are let's say a biologist, a molecular biologist, and you're working on protein X, and you would like to know the structure of protein X, you go to this database I showed you and you download the structure. If it hadn't been in the PDB, you'd download the predicted structure. You have some degree of confidence in it. In fact, AlphaFold comes with a confidence level. And if you have high confidence in it, and that's the protein you work with, you say, I got what I needed to know, I'm done. Who needs the crystallographer anyway? But if you are somebody working on trying to understand the underlying processes that make things work, then I submit that you will still be interested in the theory behind it, and you will still be working on understanding it from physical or physical chemistry principles. And it will be important to you, and I submit that it's going to be important to everybody else. The danger is exactly the danger you're alluding to. That is, that the funding agencies say, well, all the good work is being done in artificial intelligence. There's not so much equivalent work that's being done in laboratories that try to understand models or first principles. So we're going to start shunting the money to these other things. And the other thing, the other danger for this, for people like us, is that at least in Hopkins, a student comes into a biophysics group and wants to choose a mentor for their PhD, and the mentor has been working on something that is physically chemistry based, and they say, I don't want to do that, I want to do artificial intelligence, because that's what all the action seems to be. So it's a problem. But nevertheless, I think we keep going, do the best we can, because I think ultimately there will be the kind of success that just because it's taken almost a century with physical chemistry methods, it will ultimately succeed. I can't prove that. It may not be true. I may be incorrect. But I think back about the history of chemistry. The idea of constant proportions in chemical reactions. And then ultimately that led to Avogadro's number. And it took us 100 years to get there from the very first beginnings. And 100 hard years for some very smart people, probably as smart as the people in this room. And then when you finally succeed, you can explain it in an hour. So I don't know if that's what the future holds, but I work on that, and but I can afford to. So you have to be more cautious. I wish you well.
Okay, so here we come to the end of this talk. Let us give a big hand to this young man. Wait, let us give a big hand to this young professor who is with us. I won't tell you how young he is, but I'll tell you in 1947, when India became independent, the year when G.N. Ramachandran, who he has met, interacted, and written papers on Ramachandran plot, 1947, G.N. Ramachandran received his first doctorate degree from Indian Institute of Science working under C.V. Raman. And in 1947, if I'm not wrong, George was an 8-year-old boy, hanging out with his mother in Texas. So thank you, George. I hand over the mic to Chorajit, but I want you to stand here. Normally people take pictures of the speaker, but I want the speaker and the audience together. Good. Okay? So that you are not in isolation. This is for my own record. Wait. Thank you for showing up today. Go. So I hand over the mic to you.
Thank all of you.
So on behalf of Bose Institute again, so thank you, George. And I really, it's a personal anecdote or personal opinion that I don't think there is an existential crisis for people who work with actual real proteins. Because most of the, say, large proteins, you go to AlphaFold and you see strings after a few helices or strands, and you just see strings. There is no structure, basically. But there lies the mystery, as George was telling, those are intrinsically disordered regions or low complexity regions. There are business there. Okay. And philosophically, why the whole discussion reminds me of Max Planck. He said science cannot solve the ultimate mystery of nature. And that is because in the last analysis, we ourselves are a part of the mystery that we are trying to solve. Okay? So thank you, George, again. But now we have some memento for you. Our students will come over.
Hey, George.
George. George, pose for some picture.
May I open this?
Yes. এই খুলে দাও, কি আছে দেখাও। বিদেশে সাধারণত খুলে দেয়, এখানে আমরা প্যাক করে দেয়।
You have to explain to me what this line is.
So that is a book on an Irish woman called Margaret Noble, who came to India, became Sister Nivedita, worked for women, girls' education in India, worked for India's independence, was a very close friend of our founder, J.C. Bose, and that is a book of articles on Sister Nivedita, who also known as Margaret Noble, and Pasada Noble.
And she was instrumental in building up this institute as well.
And it seems particularly appropriate
And the other book is written by a 13-year-old guy who is just finished his PhD in string theory. He was from a state in India in northeast called Manipur. He had visited Kolkata, Bose Institute for a 2-week program, and he wrote a diary where he recorded science. He also fell in love with someone and fell out of love with someone within that 14 days. So it was love and science. He brought that diary to me. I was organizing a workshop, and I convinced my director then to publish a book. So it's a book about love and science. What else do you need?
Nothing else. Well, maybe a little food. The diary of the teenager's brief sojourn at the Bose Institute. Thank you very much. Thank you.
Thank you, George. So now I invite Professor Bose to end the session.
So,
to get it later, you can have a seat there or you can stand beside me.
No, John, let's go.
The floor is yours.
So I thank again, and I am privileged to present the vote of thanks, which is a formality. I will not keep you waiting for too long. First of all, I would like to convey my sincere thanks and gratitude to the organizers, the Indian Academy of Science, Bengaluru, for presenting this platform, and also Bose Institute for providing this lecture and helping in the dissemination of scientific knowledge, which is one of the mottos of this institute. At the same time, this whole thing would not have been possible without the person himself, Professor Rose. So thank you again for accepting this invitation and choosing our institute for this momentous occasion. I sincerely thank Director Professor Uday Bandyopadhyay, who, although he wanted, he couldn't be available today for preoccupation, for his constant support, guidance, and hosting this event. Words fell short of to thank the chair without his exciting, his childish, and his ever enthusiastic conduction of this whole lecture, Professor Gautam Basu. And he was noticed, he was being informed that within a very short time. This whole program was constituted within 5 days. So thank you, Govinda, for chairing and giving such life to this whole program. I thank Professor Shamal Roy and Professor Siddhartha Roy for introducing the speaker, both from a professional front as well as a personal touch. Heartfelt thank goes out to Professor Krishnananda Chattopadhyay, Krishnananda Da, Abhishek Banerjee of CSIR IICB, and my colleague Dr. Sarojit Polle of Bose Institute for their constant support, help, and guidance. I thank the registrar, Bose Institute, Professor Rajesh Shirai, who, being a scientist himself, have taken the pain in guiding the administration, and his entire team of Bose Institute admin, including but not limited to the accounts, engineering, and other areas, for their prompt action and help whenever we call for. And lastly but not the least, I thank the esteemed, all present esteemed fellows of the Academy, the entire scientific fraternity of not only Bose Institute but the neighboring institutes for accepting the invitation, being present here both physically and online. Scientific talks are never successful without learned audience like you. So I convey my thanks last, but of course again not the least, to the research scholars and students who have come here and asked so many exciting questions. So thank you again.
I just want to add that after lunch, there is a interaction session with students, and George has agreed to give it's open to all, a 40-minute or so long, more technical talk if anybody is interested. If people are interested, there will be that talk after lunch around 2 o'clock, at 3 o'clock in one of the lecture halls here. So all of you are invited, anyone, all of you, if you are interested both to interact and for a little bit more technical talk. And if you are going to hang around here, there is a nice dhaba on the other side. You can have lunch there. There is a canteen here. But if you want to hang out, you can hang out, and we will meet again around 3 o'clock. Thank you very much.